Hello everyone, in this lecture we will talk about transcription. So this is the reference book that I've used. You can go back and review this book if you want to. Okay, so let's talk our discussion on transcription. So first of all, let me ask you, what is the transcription? The transcription is the process by which mRNA is formed. But how this mRNA is formed? The mRNA is formed by using the DNA as a template. Okay, so this is the process by which a cell uses DNA as a template to make mRNA, and we will also talk about how this mRNA will be used to make the protein, but that will be for the later discussion. But for today, we'll talk about the transcription. So transcription in eukaryotics, it occurs in the nucleus, whereas in the prokaryotics it happens in cytoplasm, because as you know that the prokaryotes does not have a true nucleus. So this process occurs in cytoplasm for prokaryotes, in eukaryotic occurs in nucleus. Again, transcription, it is from DNA to RNAs, particularly it's a mRNA. And after this transcript is being made, there is also some post transcriptional modification that also occurs in the eukaryotics. Again, this process only occurs in eukaryotes, it does not occur in prokaryotes. Okay, so this is very crucial process. So once mRNA is being made, it is not a functional, there has to be some uh, additional process that is required, which is the post transcriptional modification, and we will also talk about what are these processes in later in this lecture. Okay, and one more thing to note here, that whatever transcript is being made is not the functional all the time. Okay, so some of them are made and some of them are being degraded, or some of them are stored for the future use. So the take home here is that not all transcript result in protein. Okay, so and on the right side of the image I mentioned about the theory of the central dogma here, in which it suggests that the DNA is used to make mRNA, which is the process of transcription, and translation, it is the process by which mRNA is used to make proteins. Okay. Okay, so so far we talk about the process of transcription. Now we will talk about different types of RNA. Okay, it's not like a cell has not only like a few of the not like couple of the RNA. There are so many different types of RNA that is present within the cell, and all of them have a different functions. So mRNA, as I said, it is used to make the protein via the process of transcription first, and then followed by the translation. rRNA is nothing but just the ribosomal RNA, and we will talk about the function of the ribosome in later in this video. tRNA, which we also will be talking about in this video, tRNA is nothing but it transfers the amino acid group to the growing protein. Okay, so it takes the amino acid and it incorporates to make the protein. Okay, so we will also talk about the tRNA structure and how they function. Okay, there is another class of RNA which is known as a miRNA. Okay, this RNA is actually regulates the gene expression. Along with miRNA, there is another RNA which is siRNA. It's just a small interference RNA, and as the name suggests, they regulate the gene expression by modifying the mRNA expression. Okay, and another class of RNA is known as a snRNA. Is just nothing but just a small nuclear RNA, and this RNA is important for the splicing. Splicing is a part of a post-transcriptional modification, and we will also talk about how this snRNA allow for the splicing to occur. And this splicing is very crucial process. So again, the take home is that there are different types of RNA and all of them have a distinct functions. Okay, so what are the transcription factors? Okay, now in order for the transcription to occur, there has to be some class of protein that actually initiate the process. It's not like where RNA polymerase just can come and start doing the process. Remember in the replication video we talk about the initiator proteins, they actually initiate the process for replication. Same in the transcriptional, where there are some transcriptional factors. They are there to initiate the process. Okay, and again they are initiate the transcription and they promote the transcription as well. So in the absence of these transcriptional factors, the transcription is unlikely to occur because they actually promote the transcription. And this transcriptional factor, they bind to the promoter region of the DNA. We'll talk about what is the promoter region in this later in this lecture. uh, so this transcriptional factor, they are specialized and they recognize the promoter region of the DNA where they can actually start where they actually aid the process of transcription. In prokaryotes, there is only one transcriptional factor, which is the sigma factor, whereas in the eukaryotes there are so many different types of factors present. You don't have to know all of them for the exam. There is the one that is mainly that is very important to know, which is the transcriptional factor 2D. Okay, so the take home here is that the prokaryotes has only one transcriptional factor, which is the sigma factor, whereas the eukaryotes have a multiple transcriptional factor, and transcriptional factors, they are aid in the process of transcription. Okay, again, the promoter. So the promoter, it is the site, it is actually the beginning site for the transcriptional. Okay, now the promoter do not involve in the process, in the direct process of making mRNA, because they do not have any functional gene, so they're not going to be transcribed to make mRNA. Instead, promoter aids in the process of transcription, as I mentioned, and this is the site where all the transcriptional factors and RNA polymerase bind. Okay, so without the promoter, or if there is a mutation in the promoter, this process will not likely to happen, because this is the initial site for recognition of transcriptional factor and RNA polymerase. So as you can imagine, that the promoter is very crucial for this process. And again, I mentioned promoter is the sequence of the DNA, it is not a protein. Okay, so make sure you do not confuse with this concept, because this concept is very important. Of students often get confused, that they think that the promoter is a protein, but it's not the protein, it is the DNA sequence. Okay, and as I said, they do not contain a gene, but since they do not contain a gene, they will not be transcribed. They only aid in the process of transcription. And eukaryotes has different types of promoter, like tata box, they call TATA box, which is just the tata box. There is also cat box and the GC region. Now all of these sequence are very specialized, and they also help, and they also aid in the process for transcription. In eukaryotes there are different types of promoter region, like a prigmo box and all of that, but again that's not really important to know. But just know that the promoter is the DNA sequence that actually aid in the process of transcription, and they will not be transcribed. Okay, so on the right side of the image I mentioned, I put the image for the DNA sequence here, which you can see this is the promoter region here. And let me bring my pen. So this is the promoter region, and next to promoter there is a gene that will be transcribed, but there are also some other sequence that are present that will not be transcribed. But again, just know that it's the gene that will be transcribed, not the promoter. Okay, so all of this uh, 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 promoter sequence you see here, tata box, cat box, GC, they will not be transcribed. Okay. Okay. Also, as I said, in the DNA replication, there are some initiator proteins present that actually aid in the process of replication. Similar to that concept, there is an also some sequence called enhancer and the repressor, and the name is very intuitive. You can easily guess what they will do. Enhancer will actually increase the rate of transcription, and silencer, which will decrease the rate of transcription. Again, this is a specialized DNA sequence, not a protein. Okay, I am putting so much emphasis on this concept because uh, only few changing in the few wording of the question can actually change the whole answer. So make sure you know that these are the sequences, not the protein. Okay, and since I said it's a sequence, this is the site where the proteins can bind, just like the promoter, right? The promoter is the site where the transcriptional factors bind, whereas here activator proteins they bind. So activators will bind to the enhancer. As you can see here, this is all the activator proteins that actually bind here, and they actually promote or they enhances the process of transcription. And the repressor proteins, they bind to the silencer, and again their function is to silence the gene, prevents the transcription to occur or reduces the rate of transcription. And one more thing to note here, that the enhancer and the repressor, they can be located proximal to the promoter or they can be located at distal to the promoter. So this is something that is very unique, because promoter, as I mentioned, it is located proximal to the gene. Remember I talked about on the previous slide here, right here, that the promoter, it is located at proximal to the gene, and it has to be located proximal to the gene so that the transcription can occur, whereas the enhancer and the repressor, they're located either proximal or they're located distal. Okay, so let's say if it is located at distal site, then when transcription going to happen, the DNA, the sequence will kind of looped around. As you can see here, this enhancer is located far distal to the promoter. So when the transcription is going to happen, the DNA sequence will likely to loop, and which brings the promoter or the repressor proximal to the promoter region. So this whole complex, the enhancer along with the activator proteins, they can interact with the promoter and changes the gene expression. As you can see, there are multiple transcriptional factors, mediator proteins are involved, which actually change the gene expression based on the availability, based on the cell's need, right? So important concept that they are located at the proximal or distal to the promoter region, whereas the promoter, they are always located proximal to the gene that will be transcribed. Okay, so very important concept to know. Okay, so this is just a summary slide that I may put here. As I said, this process occurs in the cytoplasm. Oh, sorry, nucleus in the eukaryotes and the cytoplasm for the prokaryotes. So this process is three-step process: initiation, elongation, and termination. So initiation, it is the process by which the RNA polymerase actually recognizes the promoter, as I mentioned, and along with the RNA polymerase, these transcription factors also bind to the promoter, and it increases the transcription. Okay, so first, this RNA polymerase. Remember, in the DNA replication, we talked about helicase enzyme that actually unwinds the DNA. Whereas here, this RNA polymerase has an intrinsic activity of helicase enzyme. So they actually bind and they unwind the DNA, and it does all the things that happen in the DNA replication. Remember, we talked about the single-stranded binding protein as well as the topoisomerases that actually stabilize and prevent reannealing and the supercoiling. So this enzyme has all the intrinsic activity. There are not any other additional proteins are required. So RNA polymerase has all that intrinsic activity, so they can stabilize and open the DNA for transcription. And then as they open the transcription, again they have the same activity as DNA polymerase, where they can actually reach the template strand, which is the DNA strand, from 3' to 5' direction, and they start making the new mRNA transcript from 5' to 3' position, from 5' to 3' direction. Okay, so right here I said elongation process. So they reach the strand from 3' to 5', and they start make the new strand from 5' to 3' direction, which is the same concept as the DNA polymerase. Also, one thing that I would like to mention here, the main difference between the DNA polymerase and RNA polymerase is that the DNA polymerase required the RNA primers. Remember, I talked about this in the replication video, where the primase adds the initial RNA primer, whereas Here, RNA polymerase, they do not require the RNA primer. Instead, they can start making the new transcript from the scratch. Okay, so they do not require any additional assistance, whereas the DNA polymerase requires some additional assistance by the primase enzyme. So elongation is the relatively same process. It just start adding the complementary bases, except it adds the uracil instead of the thymine. Remember, we talked about in the DNA structure video where the thymine was for the DNA and uracil was for the RNA. So this is the another difference between the transcription and the replication process we discussed. Okay, so uracil, again, as I said, uracil is being added instead of the thymine, and they all are complementary to adenine. So adenine, in case of DNA, adenine is base pairing with thymine, whereas uracil, in case of RNA, uracil is base pairing with adenine. The termination process is where when the RNA polymerase reaches the terminal side, or pretty much at the end of the DNA sequence, it has been signaled to uh, uh, stop doing transcription, and they are released from the DNA. Okay, and when this once they are just deassociate with from the RNA, then the DNA will again reannul with itself, and forms a double strand helix. Okay, and the newly synthesized pre-RNA uh, is then released from the nucleus into the cytoplasm in case of eukaryotes. Okay, now the initial pre-RNA that is formed, it is also known as a hnRNA, heterogeneous RNA. Okay, this is not the functional or the mature version of mRNA. That's why they call it pre-mRNA. It's not the mRNA. Some textbook also refer to as an heterogeneous RNA or hnRNA, which is just a similar concept. Okay, and in case of eukaryotes, there will be some post transcriptional modification that will occur, which actually give a rise to mRNA. So after the post transcriptional modification, hnRNA is converted into mature form of mRNA. Okay, and there are different proteins that are involved for the termination. Like in prokaryotes, there is a row dependent and a row independent, where the row protein is there to just to cleave the mRNA, pre-mRNA, and row independent is where there is the formation of the palindrome sequence, like a hairpin loop structure, which actually create the steric hindrance and for RNA polymerase, so RNA polymerase stop making transcript. Just this is just the information that you don't really have to know it, but just know that there are different types of termination signals that are present, which actually terminate the process of transcription. Okay, again, post-transcriptional modification, very important process for eukaryotes, and as I said, it only occurs in eukaryote. It does not occur in prokaryotes. And in post-transcriptional modification, there is a addition of 5' capping. There is also splicing that occurs. There is also poly A tail that is being uh, added to the hnRNA to make mRNA. Okay, and one thing to note here, which is very important, that this post transcription modification is a simultaneous process. Okay, so it is not like when the RNA polymerase is completely made a new transcript, and from there the post transcriptional modification occurs. So this process is simultaneous. So as our RNA polymerase is making the mRNA transcript, pre-mRNA transcript, it is this post-transcriptional modification is also occurring along with the RNA polymerase. So it is a simultaneous process as the pre-RNA is synthesized. Okay. Okay, so capping, which is the 5' capping. So as the name suggests, there is some additional addition of the cap at the 5' end of pre-mRNA. Okay, so there is an addition of methylguanosine at the 5' end position. So here is the structure that I've depicted here. This is the methylguanosine that is being added to the 5' end of pre-mRNA. Okay, And also that the phosphate is also present at the 5 prime end. So this 7 methyl guanosine is also added along with the phosphate that is present here, as you can see here. Okay, so they are being this 7 methyl guanosine is added along with the phosphate, so they've just formed that structure. Okay, and now you may wonder why this capping is being added, why this all modification is happening. And the reason for that is that this pre-mRNA is likely to be degraded if this post-transcriptional modification is not occur. Okay, so there are endonucleases present in the nucleus, as the name suggests. They're going to cleave this pre-mRNA, so it actually degrades this pre-mRNA. So if this modification is not occurring, that means this endonuclease can easily cleave this newly made transcript, which we do not want. So that's why the nature has evolved such that this capping and this modification actually prevent the endonuclease to degrade the pre-mRNA. Okay, and again this process is simultaneous, as I said. It occurs simultaneously with the RNA polymerase. Okay, okay, polyadenylation. So this is again addition of some adenosine molecule at the end of pre-mRNA, which is at the 3 prime end position. So let me draw mRNA. This is the 5 prime and this is the 3 prime. So we talked about the capping that occurs here. Okay, and now we are talking about the polyadenylation, which occurs at the 3 prime end here. And again, the function is the same. It prevents the degradation by endonucleases. Okay, so this endonucleases also degrades this pre-mRNA. So this polyadenylation and this 5 prime capping prevents the endonuclease to degrade the pre-mRNA. Okay, and how does this enzyme that we will going to talk about, which is the polymerase, how does this enzyme recognize that the pre-mRNA has how does this enzyme polymerase recognizes the 3 prime end position? And the signal here is that the AAU okay, followed by some nucleotide and GC content. So when the termination signal when this enzyme sees this AAU, let's say this is AAU with the dark ink here, followed by some nucleotide and GC content. Okay, so that's the GC. So when this enzyme recognize this whole portion, it will add around 200 to 300 adenosine molecules. Okay, this is just the uh, basis. They add crazily. Okay, so they add like 200 to 300 adenosine molecule at the 3 prime end. Okay, and after addition of that, it prevents the transcript to be degraded by the uh, endonuclease. It prevents the endonuclease to degrade the transcript. Okay, also these two uh, phenomena, the 5 prime capping and polyadenylation, it also aid the mRNA to be transported out into the cytoplasm from the nuclear pore. Okay, so they also do this process. Splicing. Okay, this splicing is very important concept in cellular biology, because there are many diseases that are involved if this splicing is not occur properly. Okay, so this is the process by which the introns are removed and the exons are retained. Now why it is the case? Because exons are the coding region. They will code for the specific protein, specific amino acid, whereas the introns, they do not do any of that. Okay, they are not the functional gene, but this intron is very important. Okay, it has some other functions too. So just know that these introns are not just the waste, they do have some other functions. And the exon, as I said, is the coding region, whereas the introns are not coding, that's why they are removed. And how the splicing occurs: so on the right side of the image, I put the image here. As you can see here, this is the exon, and the green portion that you can see here, it is the intron. And now how the introns are being recognized: so intron has a specialized recognition bases such as guanosine, uracil, adenine, guanine. Okay, guanine, uracil, adenine, and guanine. Okay, so gu at the 5 prime end and ag at the 3 prime end. So when these uh, bases are present, that means that is the indicative of introns. And there is a specialized protein known as the snRNP. They are also known as the snRPs. So these snRPs are very specialized protein. They will bind to this mRNA here, and along with mRNA, when this nerve protein bind with mRNA, they form a complex known as spliceosome. So this spliceosome complex is formed, which actually cleaves these introns from the exons. Okay, so they cleave these introns, and this introns that is formed is also known as a lariat. Okay, so as you can see, this is the lariat. So when this lariat is formed, and then spliceosome cleaves the introns from the exon, and this lariat is being removed. Okay, so again, this is something I mentioned. RNA to form the spliceosome, introns has a specialized sequence like gu and ag, and introns they are looped and they removed via the spliceosome complex. And when these introns are removed, then these exons are connected, and uh, exons are connected, so now they can be ready for the translation. And as I said, there are many mutations that can happen in this process. Okay, okay, alternative splicing, this is very unique concept. It only happens in the eukaryotes, and it's very important uh, because because by this process one gene can give a rise to many different types of protein. Okay, so we have one gene that can actually form multiple of different functional protein. Okay, it's a very important process because remember we have millions of different types of protein, and we don't have a gene for all of that protein. So the nature is very smart, and the nature is very smart, so it has only given a one gene that can make a different types of protein, and their function will be different too. So here on the right side, as you can see, this is just the a mature mRNA. So as you can see, either uh, they are exon 1 and exon 3 and exon 2 are connected to form a different protein, or 2, 1 and 3 they are connected to make a different protein, or they can just stay as it is to form a different protein. So this is a very unique concept, and they give a rise to a diverse class of proteins. Like, remember, we have different antibodies present, we have different receptors like angiotensin 1 receptor and angiotensin 2 receptor, ADH type V1 receptor, V2 receptors. They're just the same, they are made up of the same gene, but due to this alternative splicing, they have evolved to get the different function because of this process. So this process is very important. Okay, and so here I'm concluding the video on the transcription. If you have any question, you can put it on the comment section, and that's it for today. Thank you.